Have you heard the song by Kelly Clarkson? It's pretty.
Aww.
And it's just like, you're listening, you're like, that's a pretty tough song. And it was, there was something behind it other than, I like your booty, I like how you move. And you're like, really?
There's depth.
Right? Hi, I'm Dr. Charlotte Vines. I'm teaching 3 courses this year. One of them is developmental biology. The second one is G protein coupled receptor biology. And then the third one is mammalian physiology. We're interested in trying to control a number of different things that happen with the immune system. The receptor that we study, CC chemokine receptor 7, is involved in controlling all those processes. So what we're interested in doing is we're interested in controlling CCR7. What we're interested in having our build students do is develop tools that can control CCR7. One of the tools is a protein antagonist, and that is what you would be working with in our lab and developing during your time in the build program. Children who get T-cell acute lymphocytic leukemias, a lot of times the tumors will migrate into the brain. And there's only one way they can get in, or one way that we know of right now, and that's with the protein that we study in our lab. So if the leukemic cells express this protein, they can move into the brain. Now the reason why you care is because if the cells get into the central nervous system, they can hide. And they hide from chemotherapy that normally would be able to kill them. And then when the children come off the chemotherapy, the cells move back out into the periphery, into their bloodstream, and they kill the children. If we can successfully develop this antagonist that I'm trying to get you guys to help us develop, and we can basically save their lives after they go through normal chemotherapy treatments. So this would entail a lot of cell biology. It would entail a lot of microscope work, a lot of knowledge of basic prokaryotic cell biology, and you would learn a tremendous amount of how everything that you're learning in your basic biology fits in with research. Once you have developed some of these skills, so some of the very simple things that will teach you how to maintain a sterile field in a tissue culture lab, how to maintain sterile cultures in a prokaryotic setting, how to analyze DNA by restriction mapping. Each one of those techniques lays the foundation for very important studies that can be done and can be expanded upon if you decide to go into any one of those fields: DNA analysis, protein expression, cell biology experiments. So each one of those techniques, you're going to be able to lay foundations for a much broader, much more wide-reaching area of research. We're very anxious to have you in the lab. I think it would be a lot of fun. You're going to learn a lot of techniques that I never even dreamed of learning when I was an undergraduate. So I encourage you to come see what we're doing. Come talk to me if you still have questions, and go Miners.